Bismillahirrahmanirrahim. We are discussing inflammation, and today we will discuss granulomatous inflammation, that is in continuation with the chronic inflammation. In the last lecture, we discussed that acute inflammation is characterized by the infiltration of acute inflammatory cells, particularly the neutrophils. While in chronic inflammation, there is infiltration of chronic inflammatory cells. These are the lymphocytes, monocytes, macrophages, which make the mononuclear phagocyte system or reticuloendothelial system, plasma cells, and sometimes other inflammatory cells. So the main difference based on cellular infiltration is that in acute inflammation there is infiltration of the neutrophils, while in chronic inflammation there is infiltration of the lymphocytes, monocyte macrophages, plasma cells, and other chronic inflammatory cells. We know that whenever we talk about the chronic inflammation, we talk about the infiltration of the chronic inflammatory cells. But in this situation, we don't know that which organism or which condition has caused this chronic inflammation. And we have to use the microbiology lab and microbiological and other serological and immunological procedures to find out the causative organism. Particularly if we suspect a bacterium, then we do certain microbiological tests and culture of the specimen that we get from the patient and grow the bacterium, and then to see that which bacterium uh, has the which bacterium has caused the disease. But certain chronic inflammations are of such a type known as the specific inflammations. And the chronic specific inflammations has a specialized microscopic and sometimes gross appearance and structure that without going into the microbiologic procedures, we can assume that which type of bacterium this is. So granulomatous inflammation is a chronic specific type of inflammation which is characterized by the formation of granulomas. Now, why granulomatous inflammation occurs is like chronic inflammation. When the acute inflammatory reaction is unable to solve the problem, or in acute inflammation, if the cells, inflammatory cells, are unable to kill the invading agent, then there is a specialized type of inflammation is required. So when we say granulomatous inflammation, it is a specialized type of inflammation which occurs when the usual chronic inflammation can't get rid of the offending agent. Now, what is the characteristic of granulomatous inflammation? Chronic granulomatous inflammation is characterized by the formation of granulomas. And these granulomas have so typical and specific picture and appearance on microscopic and even sometime on gross examination, for example in tuberculosis, that we identify the disease and the causating agent. Now, what are granulomas? Granulomas are localized collection of epithelioid cells. Epithelioid cells are nothing but they are the modified macrophages, and we know that the macrophages are derived from the circulating cells known as the monocytes. The monocyte is transformed into the macrophage. The macrophage is more powerful cell to kill the microorganisms. But if the microorganism or the causating agent, bacterium or virus, is resistant and it is difficult to be eradicated, then the same macrophages are converted into very specialized type of cells. And the same is the situation that happens in granulomatous inflammation. Here, the macrophages, they are modified into epithelioid cells. And the picture that we see in granulomatous inflammation is a localized collection of epithelioid cells, which are modified macrophages. They are surrounded by the lymphocytes. Within the epithelioid cells, we can see the giant cells. The giant cells are formed by the fusion of the several epithelioid cells, and they are multinucleated. Right? Then there is an enclosing rim of fibroblasts. And if the infection is caused by a parasite, then we also see the eosinophil cells, which are one of the three cells, neutrophils, eosinophils, basophils, that are granulocytes. And here in parasitic infections, we also see the eosinophils. This picture is a diagrammatic presentation of a granuloma in which we can see the epithelioid cells. Within the epithelioid cells, there are multinucleated giant cells, which in tuberculosis are called the Langhans giant cells. The epithelioid cells are surrounded by the lymphocytes, and since it is a chronic inflammation, the healing process is also a characteristic of this type of chronic inflammation. And in this picture, we can see that the collar of lymphocytes is surrounded and enclosed by a rim of fibroblasts. Since this diagrammatic presentation of a tuberculous granuloma, so there is in the center an area of cell necrosis and inflammatory debris, and that is known as the caseation necrosis. Caseation necrosis is very specific for the tuberculous infection, which is caused by the Mycobacterium tuberculosis. Now, diseases that are caused are characterized by the granulomatous inflammation. There are 6 diseases which are the major diseases discussed in the book, and they are characterized by the granulomatous inflammation. The first one, which is known as the prototype of granulomatous inflammation, is the tuberculosis. And tuberculosis, a very common disease, it is caused by the Mycobacterium tuberculosis, which is an acid-fast bacillus. So, what do we mean by acid-fast bacillus? Actually, when we examine a specimen in the microbiology lab, we do gram staining, in which we use alcohol or some acid to decolorize the picture. But in case of tuberculosis, the acid-fast bacillus is so resistant that it will resist decolorization by alcohol and normal acids that we use in gram staining. So here we use a special type of stain known as the Ziehl-Neelsen staining. And we identify the Mycobacterium tuberculosis and acid-fast bacillus by using 20% sulfuric acid, which is a strong acid, to decolorize this bacterium. The Mycobacterium tuberculosis is grown in the laboratory in a special medium known as the Levenstein-Johnson medium, or simply the LJ medium. The second granulomatous inflammation is the syphilis. which is caused by the bacterium known as the Treponema pallidum. And the syphilis is characterized by the formation of gumas, and we divide syphilis into primary, secondary, and tertiary syphilis that we will discuss when we will be discussing the syphilis on detail. The third granulomatous inflammation is the leprosy, which is caused by the Mycobacterium leprae. And the fourth type of granulomatous inflammation is the cat scratch disease, which is caused either by the infected bite by the infected cat, and sometimes even scratch may give rise to the disease. These cats, they carry the infection, and the organism, which is a bacterium and is known as the Bartonella henselae. And the disease can be transferred to the humans by the bite of the cat, and sometimes when the cat scratches the person with its paws, so they will transmit the infection, for which the causative organism is known as the Bartonella henselae. This disease may be vary from very mild to severe, particularly in those individuals and those patients who are immunocompromised, in which severe type of infection and inflammation occur. The sarcoidosis is the fifth type of granulomatous inflammation. The exact cause of sarcoidosis is unknown, but what happens that there is an autoimmune type of reaction occurs, and the disease affects many systems in the body. So sarcoidosis is not caused by any bacterium, virus, or fungus, or any other microorganism. The fifth type of granulomatous inflammation is the Crohn's disease. And again, Crohn's disease is caused by the immune reaction against the self-intestinal bacteria, which is the normal flora that exists normally in the large intestine. Crohn's disease is one of the two types of inflammatory bowel diseases. One is the ulcerative colitis, and the other is the Crohn's disease. So here again in Crohn's disease, no external organisms are involved, but it is said that the Crohn's disease results from the reaction of the immune cells against those bacteria that are known as the commensals, and they normally exist in the large intestine without causing any damage to the person. And they are therefore also known as the self-antigens. Now, before we discuss granulomatous inflammation in more detail, let us recall the major chronic inflammatory cell known as the macrophage, which in granulomatous inflammation will give rise to the formation of epithelioid cells. Now, the monocytes, they normally circulate in the blood, and when there is an infection, which may be a bacterium, virus, or any other type of invading agent, these monocytes circulating in the blood, they are transferred from the blood into the tissues. And the monocytes and the macrophages, they form the monocyte macrophage system, also known as the reticuloendothelial system. These are mononuclear cells, and the monocytes, when they transfer from the blood into the extravascular tissue, they are activated and converted into macrophages. Now, the macrophage, before it performs its action, it must be activated. And this is the activated macrophage that will perform certain functions which play a very vital role in inflammatory diseases. Now, we have discussed that there are two ways the macrophage can be activated. One is known as the classical pathway and the other is known as the alternatively activated pathway. Macrophages that are activated by the classical pathway are known as the M1, and those activated by the alternative pathway are known as the M2. Now, classically, the macrophages can be activated by microbial products like endotoxins, polysaccharides, and also by the cytokines that are derived from the activated T cells, T lymphocytes. For example, interferon gamma and other cytokines. Interferon gamma is the major cytokine that is derived from the activated T lymphocytes and that will classically activate the macrophage. Some foreign substances, crystals, and particulate matter also can activate in a macrophage through the classical pathway. Now, if the macrophage is activated by the classical pathway, it will play its major role in inflammation. It will facilitate, it will amplify, and it will trigger its primary role in inflammation. On the other hand, when the macrophage is activated through the alternative pathway, it will play its role in healing and repair. So this is the same macrophage which play two different roles in two different situations. The classically activated macrophage will produce reactive oxygen species, reactive nitrogen species, proteases, certain cytokines, coagulation factors, and arachidonic acid metabolites. And all these play their role in inflammation. And the major purpose of the classically activated macrophage is to kill the microorganism. As we know that reactive species and enzymes, various cytokines, they also are used in killing the invading microorganism. But the same macrophage can be activated through alternative pathway in which the cytokines interleukin 4 and interleukin 13, which are released from the activated T lymphocytes, the mast cells which are located in just in the perivascular tissue around the blood vessels, and which are the major source of histamine, and the eosinophils, which are the major cells that play a role in allergic reactions or hypersensitivity reactions, and also in parasitic infections. So the cytokines interleukin 4 and interleukin 13, they are released by T lymphocytes, mast cells, and eosinophils, and they will act and they will activate the macrophage through the alternative pathway. Now, the macrophage that is activated by the alternative pathway will produce certain factors that will help in tissue repair and fibrous tissue formation. For example, these alternatively activated macrophages release the platelet-derived growth factor. They derive the fibroblast growth factor. They they secrete the transforming growth factor beta. Some fibrogenic cytokines and angiogenic factors that are involved in the formation of new blood vessels to the newly formed fibrotic tissue as a result of repair and healing. And they also produce certain collagen remodelers because the fibroblasts, they will secrete collagen, and fibroblast and collagen synthesis, it is an important component of the healing process. Now, acute inflammation is the acute response, and what happens in acute inflammation, that the macrophages, when they kill the microorganisms, they disappear. How they disappear? They either die by the apoptic cell death, which is the programmed cell death, and in which a cell undergoes suicide. We have discussed the apoptic cell death in detail when we were discussing the cell injury. And sometimes the macrophages that are not needed more here in acute inflammation, they will make their way to the nearby lymphatics. So it is clear that in acute inflammation, when the inflammatory process is resolved, when the microorganism or other invading agent, if it is killed, then the macrophages will disappear from the scene. But the situation in chronic inflammation is a little different. Here, the invading agent, the microorganism, the bacterium, the virus, it is very resistant to be killed by the macrophages. And the macrophages are supposed to stay here. And in chronic inflammation, the macrophages tend to accumulate at the site of inflammation because their action is needed more and prolonged due to the resistance and due to the resistance of the invading agent, which is more powerful and strong. Now, this accumulation of macrophages in chronic inflammation is the result of continuous recruitment of the macrophages from the blood. Monocytes, they pass from the blood into the extravascular tissue and are converted into the macrophages. But here this process is continuous. Sometimes local proliferation of macrophages at the site of inflammation, which we can say is a type of local clonal expansion or local expansion, in which the macrophages proliferate and form a large number of mononuclear macrophages to fight against the invading agent. And in chronic inflammation, the macrophages are immobilized to go into the lymph nodes and to stay at the site of the chronic inflammation. And this immobilization of the macrophages is caused by the secretion of migratory and inhibitory factors by the activated T lymphocytes. They don't allow the macrophages to leave the battlefield. Here, one thing important that I want to say, that in addition to killing the microorganisms and in addition to removing and degrading the necrotic tissue, the inflammation, the macrophages, they release reactive species in the form of reactive oxygen species, reactive nitrogen species, and enzymes known as the proteolases, which include various types of hydrolytic enzymes. And in addition to killing the microorganisms, potentially these components of the activated macrophages can cause severe and extensive damage to the normal healthy tissue. And this damage sustained by the normal healthy tissue is a characteristic of chronic inflammation. Because in acute inflammation, usually healing occurs through resolution by restoring the normal anatomical and physiological integrity of the tissue. But whenever there is chronic inflammation, there will be healthy tissue damage, which may be mild or very severe. Now, coming back to granulomatous inflammation, which is a specialized type of inflammation which occurs when the normal chronic inflammatory response is unable to kill the offending agent. The circulating monocytes, they are shifted into the extravascular tissue to the site of the inflammatory response, and they are modified into macrophages, and the macrophages are further modified in granulomatous inflammation to epithelioid cells. Now, in this picture of granuloma, we can see the localized collection of epithelioid cells. Some giant cells are also present, the epithelioid cell uh, collection is surrounded by the lymphocytes, and outside the lymphocyte collar there are, we can see, fibroblasts. Now sometimes these epithelioid cells, several epithelioid cells unite to form the giant cells. So the giant cells are formed by the fusion of several epithelioid cells, and therefore they are multinucleated. Now here we briefly discuss the content cells of the granulomatous inflammation. Epithelioid cells, as I told you, that they are the modified macrophages which are derived from the circulating monocytes, and they have got a pale eosinophilic cytoplasm. Eosinophilic, we mean that it during histopathological examination they will take the eosine stain in the HE stain, hematocrit eosine stain, which, and the eosine stain is uh, a pink color. So they, epithelioid cells, they will take the eosine stain and they will appear as pale pink color to in their cytoplasm. And in epithelioid cells the chromatin is dispersed, and this dispersed chromatin indicates that the epithelioid cells are more synthetic than phagocytic. They synthesize various cytokines, and the nuclei of the epithelioid cells, they are elongated in a foot-shaped appearance, and the phagocytic capability of the epithelioid cells as compared to macrophages and other phagocytes, it is less. But as I said, that the macrophages have strong secretory ability, secreting various cytokines, proteases, reactive oxygen species, and other components of the inflammatory reaction. And as compared to macrophage, the epithelioid cells are more strong in their power to kill microbes. The giant cells, they are formed by the fusion of several epithelioid cells, and there are two types of giant cells. One is the foreign body giant cell, in which the nuclei, they are scattered in the cytoplasm, and the other is the Langhans giant cells, particularly seen in tuberculous infection, and here the nuclei are arranged in a collar at the periphery and form a ring in the cell. Now, morphological patterns of granulomatous inflammation. In tuberculosis, we see the caseating granuloma, in which there is a central zone of caseation necrosis, and which is characterized by the cheesy-like appearance, and particularly during in the diagnosis of tuberculous infection, if we suspect that a lymph node has got infected by the tuberculous infection, and even when we cut the lymph node, we will see a soft, cheesy lymph node. And here we can say that even by gross examination, we can say that the infection is caused by the Mycobacterium tuberculosis. In addition to tuberculosis, in cat scratch disease, a suppurative necrotizing granuloma is formed, and it is not like the cheesy-like appearance that we see in tuberculosis. But here, one important difference as compared to other granulomatous and chronic inflammatory diseases, that in cat scratch disease we see lot of neutrophils, which are basically characteristics of acute inflammation. But since cat scratch disease is characterized by the inflammation, by the infiltration of the neutrophils, and they cause suppurative necrosis of the underlying tissue. We divide granulomatous inflammation into caseating necrosis, or necrotizing granulomas, and non-caseating, in which there is no formation of necrotic type of suppurative or caseating tissue. So the three granulomatous diseases are non-necrotized, and these are the sarcoidosis, leprosy, and the Crohn disease, and in which, unlike tuberculosis or Caseation disease, there is no central area of caseation necrosis. In this picture, we can see the typical tuberculous granuloma, uh, in which there is a central acellular eosinophilic area of necrosis, and we can also see the Langerhans type of giant cells and the blue colored lymphocytes, and the epithelioid cells. They are just like epithelial cells, and they are not very clearly seen in low power, but can be identified in high power, and they, since they are transparent like epithelial cells, so they are not so prominent as the giant cells or lymphocytes. Here is a typical granuloma of tuberculosis, known as the tubercle, showing the central acellular area of caseation necrosis, and the epithelioid cells and the giant cells and the blue colored surrounding the tubercle are the lymphocytes. This is also a picture of tuberculous granuloma, in which the central area show caseation necrosis. And finally, we conclude this lecture by simply making a summary of what we discussed. Tuberculosis, which is caused by Mycobacterium tuberculosis, and which is characterized by localized collection of epithelioid cells, which are nothing but modified macrophages, surrounded by lymphocytes. Outside the lymphocytes, there are fibroblasts, and in the central area, there is an acellular necrotic area, which is known as the caseation necrosis because of its cheesy-like appearance. Leprosy is caused by Mycobacterium leprae, and here the acid-fast bacilli, they are found in macrophages, and this is non-caseating granuloma. There is no caseation necrosis. The syphilis caused by the Treponema pallidum, a bacterium, and it is characterized by the formation of gumas. In syphilis, we discuss primary, secondary, and tertiary syphilis, and these gumas are so specific that even by gross examination, we can say that this is syphilitic guma, and in which we see histiocytes, which are the macrophages found in the lymph nodes and spleen, and sometimes plasma cell infiltration occur, and the plasma cells are derived from the B lymphocytes that secrete antibodies. That Caseation disease is caused by the Bartonella henseli, and it form around it are stellate shaped granuloma, and again there is central area of suppurative necrosis, and one of the characteristic of the Caseation disease is that there is infiltration of the neutrophils, which are seen in acute inflammation. The sarcoidosis is an autoimmune disorder, and we don't know that what triggers this disorder, but it is here also the non-caseating granuloma occurs with abundant activated macrophages. So sarcoidosis, Crohn disease, and the leprosy are characterized by the non-caseating granulomas. The Crohn disease, which is also an immune reaction against the intestinal bacteria, which are commensals and the normal habitants of the large intestine. Here also non-caseating granuloma occurs, and there is dense inflammatory exudate characterized by the macrophages, lymphocytes, and plasma cells. Thank you very much.